All right, hello everyone. So we're going to walk through the DNA replication, kind of a demo for the DNA replication interview, and talk about some of the tricks that might help you with the mural. There are going to be lots of moving pieces here, and this interview is going to take a little bit longer just because of all those moving pieces and the things that you are going to have to demonstrate the actual processes of. The first thing I'll have to say, and probably the biggest thing in terms of the tricks, if you notice here, I have my DNA structure, sugar phosphate backbone, and the bases across the middle. And what I've done, I've actually taken a whole section of this and made it a group. All right, so two different groups. So essentially, I have like four different groups here. I have two little end caps that help me keep things lined up, and these sections across the middle, which I use to unzip when I'm creating my DNA. Okay, so use the grouping mechanism there to really help you, move things around as one full unit. Okay, so let's walk through this interview and take a look at the process. Okay, so I put everything over here that we need. So we have three main categories: initiation, elongation, and termination. So with initiation, really what we're looking at is trying to get the DNA ready to duplicate and ready to replicate. Elongation is where we lay in the new DNA, and then termination is where we actually finish it up, put it together, and make sure everything is looking good. So let's start first of all with initiation. All right, so we have our DNA molecule like we've studied before. Sugar phosphate backbone and bases across the middle. And what we're trying to do is replicate this. And the reason why we need to replicate DNA is to be able to give a complete set or a complete copy of DNA to every cell, right? So the reason we replicate DNA is because cells divide, cell division. And since cells divide, we have to constantly replicate the DNA as well. Connective tissue, epithelial tissue in particular, go through replication on a regular basis. In fact, several billion times a second, your cells are dividing, and so not a second, a day, several billion times a day, and therefore you're going to have to be able to replicate your DNA on a regular basis as well. So let's look at the mechanisms involved. During elongation, there's really three different things involved. There are these things called single-stranded binding proteins. There is this thing called a gyrase, and this called a helicase. Right? These are enzymes. This is just a protein. These are enzymes that do an actual work of the body. Right? So these are basically chemical catalysts that make something happen. You can think of them as little workers, little machines. However, it helps you kind of remember that these are the things doing work in the body, right? And we talked about that the second week of school when we talked about proteins, where we're just going into a little bit more detail now. All right, so these two kind of work together in unison. So the gyrase and the helicase, they come in, they do two things. The gyrase is typically going to be kind of what unwinds and organizes the DNA. So you can see we have this double helix coming in. And we need to be able to get to the bases inside, so we're going to have to be able to unwind that DNA. The helicase then is coming down the middle, and you can kind of see the scissor shape here. It's going to actually split apart the DNA into two sections, right? And that's why I grouped these two together so that we can kind of demonstrate that all at once. We are able to unzip, like a zipper, the DNA across the middle, okay? This is called the replication fork, where this actually happens. It's like a fork in the road is where the DNA actually opens up. And to kind of take that further, if you look at the entire DNA molecule, we create what's kind of like a bubble, because typically DNA replication doesn't happen from one end of the DNA to the other. It happens along the middle of it, different sections. And the reason for this is to speed things up, right? So if DNA could only replicate from one end to the other, it would take a much, much longer time for that to replicate. And of course, like we said at the beginning, we need to do this quickly and consistently. So replication fork gets created where that helicase is unzipping. Okay? And then the final part of initiation is these things that come in. These are the single-stranded binding proteins that come in and attach to that DNA that's open and allow time for the DNA to be replicated. So they're keeping it open, not letting it wind back into its double helix form, and they just help keep it open. Okay. All of this is just the initiation. This is just getting ready to copy the DNA into two new strands. Okay. The next thing that happens then is the second part, which is the elongation phase, and this is where we're going to actually lay in new DNA. So the first part of elongation is this guy called the primase. This is kind of like when you put primer on your walls before you paint, right? You need something down first in order to actually get the paint to stick. And so the primase kind of works in this same way. The primase comes in, is going to lay a little piece of primer into the DNA. And what you'll notice is that the primase is actually RNA primase, right? So it will come in and insert a primer into the DNA, and this primer is RNA, right? So we're going to have to deal with that later. But for now, this is really just signaling where to begin the process of DNA replication, right? And so we know that we can now begin DNA replication from this point. It's going to do it on both sides, so we had opened up on this side. It's also going to, put it in down here as well, connect it into its starting point, right? And that's determined by this primase molecule, the enzyme. All right, and then finally, not finally, but this is kind of the main player in terms of putting in new DNA. But we have DNA polymerase. And what that DNA polymerase is going to do, it's going to find that primer that the primase laid, and then it's going to insert new DNA complementary to the strand that's actually there, starting at where that primer is located. Now, here's where it gets a little tricky, though. All right, so if you remember when we talked about DNA being a directional molecule, this is the 1 carbon, 2, 3, 4, 5. So this is the 3' end of the molecule, and this is the 5' end of the molecule here. Remember, we can only insert new DNA into the 3' end. All right, so the 3' end would be this side. So as that polymerase reads this existing DNA, is going to take that and inserts in this direction, 3' end, every time. So the existing DNA is technically going from the 5' to the 3' end direction, so that's why we say we're creating it from the 5 to 3' direction, but we can only add new DNA into the 3' ends, right? A's the T's, C's the G's, because we need an identical copy. We don't want any mutations at this point or changing of the letters or any mistakes, because then we get mistakes in our proteins and we can get disease and all sorts of bad things can happen. But this DNA polymerase essentially just follows right in behind this helicase as it unzips and inserts new nucleotides just like that. Okay, so this strand here is what we call the leading strand because it's going in the same direction as our helicase here. Now, like I say, here's where it gets a little complicated with the direction of DNA. This one, the polymerase is going to come down, find this. Well, I'll leave this one here. If I hit another trick to this, if I hit Control D, remember you do a quick duplicate. So it's always an easy way to make a couple extra things that you need. So it's going to come in and find this primer, right, that we've laid in from the primase. And when it finds this primer, it's going to find that 3' ends and then going ahead start laying in new DNA. But if you notice in this case, it's this direction that we're going to be laying in new DNA. It's the opposite direction of our helicase, which is going out towards the right there. So we are going to take this and let's see, go for this one here. G's to C's, A's to T's. And so that's all the same, right? There's nothing crazy there. We're adding to the 3' end. But if you're kind of paying attention, you're noticing here that we're getting a bit of an issue. What have I just done? Sorry. We're getting a bit of an issue right here. Cool. With this section here because it is empty. Let's try to stop being fancy here. All right. So again, the DNA polymerase reads it and lays in. All right. But that issue occurs when we have this section here. Since we can't add to this side of the primase, how are we going to fill in this section of DNA? Because the helicase is continuing to unzip it into that left to right direction. Okay. So what has to happen is the primase has to come in again, and it has to another piece of primer. It has to lay in another piece of primer. All right, just like that. It connects it, right? Showing where to begin again, and then it can take off. And then we are going to continue the process of, not A to T, like that. Okay. Again, the polymerase finds that primer and begins the reading in that 3 to 5 prime direction, or you can think of it as adding to the 3 prime ends, whichever is easier for you to understand, just like this. And like I say, this interview is going to take a little longer just because you're going to be moving a lot of stuff around, and that's fine. We're going to spend a little bit more time with this, and it's a little bit more difficult of a topic than the simple DNA structure that we looked at. Okay? So as that DNA polymerase reads, it's inserting with our complementary base pairing all of our different sections of DNA. Okay, just like that. All right, so what we have here then are a bunch of pieces, all right? This strand up here, because it is going in the same direction as the replication fork, is called the leading strand, and this strand here is called the lagging strand because it actually is inserting new DNA away from that replication fork or away from the direction that the helicase is opening. Now, these fragments are called Okazaki fragments, right? So it's a fun name to remember, but we have a bunch of pieces that we're going to have to deal with before we're going to be able to finalize this DNA, right? But this is really the elongation phase of the DNA replication. We have, you can notice here, we have another double-stranded piece of DNA, and if you compare this one to this one, they're going to be identical, right? GC, GC, TA, TA, GC, CA. So it should be identical. If not, you can have some issues. Right. The third part to DNA replication is this termination part here. And this is where we're going to have 2 different enzymes called endonuclease and ligase, right? And they're going to help finish things up. So the endonuclease comes in. We'll start here in the leading strand. And basically just removes the RNA that's in there because we don't want to leave RNA inside of our DNA. And it is going to insert DNA in its place, right? So we get rid of that primer and insert the DNA. Right? And it's going to do the same thing there on the lagging strand. It's going to find those pieces of RNA, remove them, and then insert the appropriate DNA. Right? Oh, look at that. There's one more. It finds it and pulls it out and then inserts DNA. Okay? So now we have 100% DNA thanks to the endonuclease, but we still have one issue left, is that we have all of these breaks because of our Okazaki fragments, right? This is not a continuous molecule, and we need that to be continuous, right? And so this final enzyme comes in, finds where those holes are, and I forget exactly where it was, but we'll put it right here, and we'll glue together the sugar phosphate backbone by inserting a phosphate group, creating one continuous DNA molecule along the way. Right? So once that's happened, then we can release all of our different enzymes. Everybody goes off to find another piece of DNA to do some work on. The single-stranded binding proteins can release now because we don't need this thing to be open any longer. It can twist back down into its double helix. And then that is that. So this is 2 brand new strands of DNA. This process will continue. We could take this on forever if we wanted to show the length of the DNA. But what you'll notice here is that we have 2 brand new strands of DNA, half of which is original and half of which is new. Right? So this half was the original, this is the new half, original up here, and then the new half here. All right? Now, one copy can go to one cell during cell division, the other copy will go to the other during the process, like we talked about with mitosis, right? With somatic cells, at least. All right, and this is the process of DNA replication. I think I showed you all the tricks. Again, Command D does some quick duplication. You can rotate a group now if you need to rotate something in order to make it fit. So you could, rotate the whole nucleotide or whatever you need to do. But that should pretty much do it. All right, thanks for listening.